Thank you. Well, the fact you stayed until now, it means you actually want to hear the story. So I appreciate, or that you want to hear the speaker that comes after me. But I apologize for the delay. And today I'm going to talk to you about. Today I'm going to tell you about the effect of the COVID-19 proteins on the vascular permeability. Thank you for inviting me today. And for me, it's always a challenge to present after Noam. It's like Noam is doing amazing work, and thank you, Noam, for presenting it and for being the chair. I'll do my best, especially when it is so late today. So, we heard about COVID all day long, so I'm not going to tell you how shit it is. The only thing I want to remind you that although it was only like 3 years ago, we basically forgot how it was at the beginning. So I just want to remind you that at the beginning, when COVID just started, we thought it's basically zombies in the street and we're all going to die, okay? Just to remind, just the way it was, I went down to the garden beneath my apartment, and the police came and told us, you need to go back to your apartment, it's locked down, and it was horrible, right? So this is how it was at the beginning. And as all the labs, we wanted to try to contribute with what we can do. And basically what we can do in our lab is to work with advanced in vitro models, human models, in vitro. This is what our lab do. What we do in the lab is basically bringing organs on a chip, specifically brain on a chip. We do sensors in chips, and we can link different organs. And basically this is what we do in the lab. So we said, okay, let's try to harvest or to take harness our lab capabilities and try to contribute something to this pandemic that nobody knows anything about it back at that time. So, okay, so we want to help fighting COVID or to understand a little bit more in COVID, but actually what can we do? It just started. So we started to brainstorm and we said, okay, let's think about it. And we said, okay, we cannot work with the virus. Back then, it just started. You need to have this BLS 3+++ to work with it, and there was only one place or 1.5 places at the time that you can actually work with the real virus. So it's not really easy to get it. You need to have something. All the people talked about ACE2, human cells. It doesn't work with any rodent cells, anything. So it need to be human specific. And there were some indications that it can also affect the vasculature, the human vasculature. It was at the beginning. Today we know much more. At the beginning we didn't know anything. And we said, okay, the vasculature is interesting, but what is currently known? So for example, in March 2021, there were already 100,000 papers about COVID, which is crazy. Today there are already above 200,000 papers about COVID. But when you look at the vasculature, there were only 12. Okay, from all these 100,000. So we said, okay, we will try at least to do something with the vasculature mechanism with our lab. So the first thing we did was to assemble, So the first thing is to think about the approach. And with all the limitations I told you, our approach was the following. So we know that the COVID actually affects somehow the blood. We know that we didn't know exactly which effect, but we wanted to see the blood. So for this, we decided to work with the human endothelium. And the protein, as we saw, there are basically 29, the virus has 29 proteins. And we said the following approach. Let's take each one of the 29 proteins and let's see which one of these proteins has the most significant effect of the vasculature. Obviously, it would be nice to work with the whole virus, but it was impossible at that time. So the following approach was that we will get plasmids for the 20, that each plasmid can basically encode for one of these 29 proteins and to identify like this, the most significant one on the vasculature. So the first thing you do is obviously to assemble a team. So we collaborated on this project together with different people from our lab. We had Rosanna, she was our postdoc at the time, and Yael, our research assistant, and obviously these terrific students. We collaborated with Uri Asheri's lab from neurobiology, with Roded Sharan from computer science, and from Kobi Nachmias and his team from the Hebrew University. And we also contacted many good people like Eran Bachrach, who wanted to help us at the beginning with his virus, and many other people who really wanted to. It was really a team effort at that time, or until today. So, as I said, I will show you our approach, what we basically did with these 29 proteins. The idea is that we culture human endothelium on a transwell, and then we want to check the following parameters. We wanted to check permeability. We do it with a concept known as TER, trans-epithelium electrical resistance, which is basically 2 electrodes that you measure the resistance between them. We did immunohistochemistry on the vasculature to see how it is affected. And we looked at cytokine release, specifically these two, the TNF-alpha and IL-6. So basically, let me show you an example. You come with these 2 electrodes and you measure the resistance. This is the control, regular UVAC without any drug or protein or whatever. And then we started to add, for example, GFP, since we want to make sure that the virus actually, or the protein was expressed. So we do it also with GFP as a control. You see there is no significant change. And then we start to add the S protein and all the different proteins. And you can see that there is, for certain proteins, a really significant effect on the permeability, which means that we can really identify which are the most dominant proteins that has the most dominant effect on the vasculature permeability. And here we can also do it as a heat map that you can see this is a good permeability, that basically the cells are untreated, and you can really identify which of the proteins have poor permeability. And we can also do immunohistochemistry for certain tight junctions protein and to see how they affect or how also they are affected. The next thing we checked was to see about coagulation factors, because it was shown, and we collaborated also with Galia Rav that gave a terrific talk here at the morning, that basically we saw that there is this dual effect. From one hand, the vasculature becomes more permeable, but from the other hand, we see increasing coagulation. So what we stained is for VWF factors, which are basically upregulated during, they show you have upregulation when there is a coagulation. So we were able to quantify, for example, the untreated, the GFP, and proteins that we saw that are significant. So here you can see 3 of the proteins that we saw that are significantly increasing the upregulation of coagulation factors. We looked at the upregulation of cytokines, and we were able to identify which of the COVID proteins have the most significant on the cytokine upregulation. And then we said, well, it is cool that we can actually see all these parameters on UVACs, but wouldn't it be really nice if we can not only describe what is going on, but actually to have a tool that we can predict what will happen? This will be super awesome, right? So actually, we found this amazing prophet, which is Oded Sharan from computer science. This is a real picture I took from his office. No, I'm kidding. So, and basically what he did with these tools from computer science, he developed this protein-protein interaction algorithm that can actually predict or identify which proteins can be affected from a given protein. Okay, so let me give you an example. So we know that we measure changes in functionality, right? So we measure the permeability of the endothelium, and we saw a change. Now, by doing biology, literature survey, consulting with people, I can tell you exactly which proteins are relevant to these changes in the functionality. Okay, specifically changes in permeability, I know it's tight junctions protein, and I know exactly which tight junction protein are related to these changes. So I can give to Oded all the what are the tight junction protein which are related to the changes in functionality. He will do his magic with the protein-protein interaction maps, and he will be able, by using his model, to tell us exactly which one of these proteins will have what kind of what sort of interaction with these proteins that will have the changes in the functionality. Okay, was I clear enough? Yes? No? Thank you. So basically he developed this model that he can look on each one of these proteins, how it is interacting with each one of these proteins, and predict the changes in the functionality. So let me give you an example. For example, if we take this protein ORF9C, he can look exactly how it is interacting with each one of these protein tight junctions protein, okay? Where you have the greenish or the yellow color, it means that it's high interaction. It can also tell us what kind of interaction, not only if it is upregulating, also if it's downregulating, but here it's just a score. And you can really see a nice interaction with some of them. And we can do it with other protein. For example, this is the ORF9b, and you can see that there are different type of interactions with different type of tight junctions protein. And obviously we can do it for more proteins and to all the proteins, and like this we can have this really nice heat map that tells us which tight junction protein will be the most affected by which of the COVID proteins, okay? Perfect. So he developed this tool, and this tool is really awesome, but a tool is not good without validation. Right? So he gave us this tool and said, look, this is what I can predict. We said, let's see if it actually fit to our experiments. So what we did was the following thing. We looked at specific proteins that we saw that gave a really high score with his network. For example, these three, the NSP7, NSP5, and NSP2. And we wanted to validate. In what sense? We wanted to see if we really see change in the protein expression, okay? So for example, he predicted with his model that NSP7, NSP5, and NSP2 will have a significant effect on the catenin beta, okay? So we took the control, we took the cells and transfected them with the virus, and we saw that actually his prediction worked well. We see that these three proteins have a significant effect on the expression of this specific protein, okay? He did it for another, and we did this validation for another protein. You see here, it's Coagulation 5. And again, we did immunohistochemistry, we quantified everything, and you see that this NSP5 has a significant, like these three proteins, the NSP2, NSP5, and NSP7, actually have a significant effect on the Coagulation 5. And we did this validation for many, many proteins. I will not show you the whole proteome validation we did, but actually, we were extremely happy that what he predicted happened in the lab, okay, in the experiment. But it was more than that, because what he can actually do with his model, he can actually tell us the sum of the up and down regulation, how it will affect the permeability of the vasculature, which means, look at this protein, for example. He can tell me exactly not only if each protein will go up and down, but what will happen as a sum to the vasculature functionality. And for example, if here we plot the values we got from the tier, so he predicted that this protein will have an effect that the total permeability will be this value. And basically he can calculate the permeability or give us an estimation of the permeability for each one of these proteins. And what we saw that there is a really, really nice correlation with his prediction, which is here, with what we measured at the lab, okay? So to make a long story short, we did a validation that not only predicts the protein expression, but also the change in the functionality, okay? I will finish shortly. Then we said, well, the endothelium in the human body is not only UVEC, it's more than that. There are more than 9 types of endothelium. So basically, can we apply his model to other type of endothelium? To do so, we created this massive table, which basically demonstrates which proteins are being expressed for each type of endothelium. So this is umbilical endothelium, this is pulmonary endothelium, umbilical artery endothelium, and we looked at all the different types of endothelium, and we did this huge table of which protein is being expressed. We gave this table to Oded and told him, now we know which protein interaction you need to look at, and he was able to give us a prediction of which proteins will have the most significant effect on which type of vasculature, and we were able to identify these 2 types of vasculature. And lastly, we said the following thing, and this is almost my last slide. We said, well, if we can do this, apply this model on different type of vasculature, can we apply this model on different type of tissues? And what we did was to look at the literature, which proteins we know that are being affected in the different tissues in the body, gave it to Roded, and he was able to create these huge heat maps for the COVID proteins, how they affect different proteins for different tissues. For example, for cardiomyocytes, for anthracytes, for lymphocytes, and many, many more tissues. And the super cool thing was that what we saw, again, almost 2 years ago, was that the most affected tissues will be the endothelium, but also the lung and the brain. And today, 2 years after, we know that COVID affects the most the brain, the lung, and the vasculature. Also intestines, but for me, when I prepared this talk, and I went to the literature, what is known today, I was super happy to see that actually a model we developed almost 2 years ago was able to correlate very, very well with what happened actually today in the clinic, okay? So this is basically the story I wanted to tell you, which basically we were able to identify the specific proteins that actually increase permeability of the vasculature. We were able to develop a protein-protein interaction map that can actually predict which proteins are being most affected from the virus, but also to look at their functionality. And we were able to apply this model to different types of tissues in the body. And this work was published in eLife about a few months ago. And although we were not funded by the center, we really appreciated the support the center gave us, and we added even the center in the acknowledgements. Just small publication. If you're interested in human relevant tissue, we established IPS, core differentiation facility, which can serve all of you. But this is a side note. These are the different things we do in the lab, which most of the things we do is not related to COVID. This is our team that I would like to thank for, and the funding agencies I would like to thank for, our collaborators, obviously, and to thank you for staying at this time after a very long day to listen to what I have to say. So thank you very much, and any question you might have, I'll be happy to answer. Thank you. Yes, no?
There's another disease, I'm not sure if you heard about it, called cancer.
No? Is it a one type of, or there are multiple?
There's a lot of vascular changes there.
Yes.
Are
yes.
you going to apply what you did there to the other disease as well?
So I was extremely interested to see the lecture that was before mine, to see if I collaborate with this speaker about doing bioinformatics and these kind of things. But seriously speaking, the easier thing to do with this is that I was able to decide relatively easy what is the parameter I want to check about the permeability. And cancer, there are so many factors that are being released and changed that I'll be happy to brainstorm about what type of factors to look how we change the permeability, because we know there is vasculogenesis, we know there is all these factors that actually, for example, glioblastoma, you know that the permeability is extremely high. There are many factors actually that cause this permeability. So if there are specific factors that you are aware of that worth checking, definitely we can use this model to test this. Okay. Oh, yes, sorry.
Hi. So do you know the relative weight of the protein-protein interactions in the decrease that you see? For example, have you weighted in differences in RNA or protein synthesis?
Yes.
Or degradation? Or what is the relative contribution of direct protein-protein interaction?
It's a super relevant and good question. Yes, the short answer is yes, we weighted, and I can show you, like, we have tables of exactly how much we give to each one. But the longer version is that since I was limited with time, I decided to not to put so many tables and graphs. But yes, we have, and I can show you, share with you, and
Part of it is that they could be undergoing, for example, de-discrimination.
Right.
From endothelial
Definitely. You're completely right. And it is something we need to take into account. Obviously, there is limitation how much we were able to take into account, but definitely it was one of the parameters that we looked at in this specific protein-protein interaction maps. Definitely. Thank you for the question. Okay, thank you, everyone.